want to continue our discussion of the cell cycle. So remember, in the cell cycle, there's the G0 phase. This is where the cell is doing nothing. It's just kind of chilling out. It's not really doing anything, at least nothing in terms of dividing. Then it will go into the G1 phase. This is where it's going to start amassing what it needs in order to divide. So getting supplies. It'll go into the S phase. This is where it's going to synthesize DNA. So basically make a copy of its genome. Now, during this phase, also the centrosomes are going to get duplicated. And then we go into the G2 phase, which is the second gap or another growth phase. And during this one, the cell continues to grow and it makes copies of organelles. It also makes ATP because it's going to need energy to divide, and it makes proteins that it's going to need once it divides. Now, what that means is by the time we're here, we have a copy of DNA, we have a copy of our organelles, and so now we're getting ready to divide. And that is where we get into mitosis, which is sort of the central thing that I want to talk about in this video. When we think about the phases of the cell cycle, we can divide them. And there's a very simple mnemonic that I usually tell students, which is I passed my anatomy test. Now this is a little bit more relevant because I typically teach anatomy, but you can think of perhaps another mnemonic that would work for this. The I is for interphase. That is that G0, G1, S, G2 thing. So at the end over here, we've got basically copies of everything. Then we get into prophase. And we'll talk about that in a minute, but I want to name all the phases. After prophase is metaphase. After metaphase is anaphase. After anaphase is telophase. And then there's a final phase called cytokinesis. Often this gets lumped with telophase, but we'll actually talk about why it's important to think about them as separate phases. Let's think of where we are. When we finish interphase, our cell has basically twice as much DNA as it had before. It's been replicating that DNA. But because we've been copying the DNA, it's unwound. It's kind of everywhere. So we have this like mess of DNA. What we need to do in order to get that cell ready to divide is we really need to pack that DNA. So this is where we're going to move into prophase. And during prophase, we are going to pack that DNA into those sister chromatids, so basically into discrete elements. The next step of moving, and this is also going to happen during prophase, but this is more the pro-metaphase portion, is we're going to dissolve the nuclear envelope. Now, if we think about this, this makes sense because basically what you're doing is you're going to be moving these sister chromatids apart, and you want half the genetic material to go to each side. Since that's what you want to have happen, you can't have any walls in the way, right? So we have to break those walls down. And to move everybody, we really want to get them organized. And what this does is this means that another structure is going to arise. So in those centrosomes, we're going to have these spindle fibers coming out. And you've got two centrosomes, one on either side. What's going to happen is these centrosomes are, through prophase, going to move to opposite ends of the cell, and they're going to start attaching to those kinetochores. Now, the important part of that is that means that they're going to be able to move around those chromosomes. When we move to our next stage, which is metaphase, we're going to move everybody to the middle so that we can take half on each side. I always think of that M in metaphase as being for middle. So a cell in metaphase would have all of its chromosomes lined up down the center. And all that lining up has been done by these centrioles that have kind of pulled everybody so that they're lined up in the middle. Now, these spindle fibers are attached on the kinetochores. So what's going to happen next is they're going to start to pull. And when they start to pull, we're going to end up in a phase called anaphase. And this is going to pull those guys apart. In fact, I always think of the A as being for apart. Okay. Once it pulls, it's going to start pulling. Now we're going to end up with the separating of those sister chromatids. And the pulling of those sister chromatids to the opposite end of the cell. Once they're on opposite ends of the cell, now where those chromatids are going to start to decondense. And this happens in a phase called telophase. And basically, what's going to happen is we're going to end up with little messes of DNA that look not unlike what we had in our original cell. But we're going to have two of them. And during this telophase portion, we're also going to develop a nuclear envelope again. And those spindles are going to break down. So during cytokinesis, what's going to happen is our cell membrane is going to start sort of pinching in. And at the end of cytokinesis, we'll actually have two copies of the original, or two cells that look exactly like the original cell. So let's take a second and think about these phases and why they're important to our goal. And remember, the goal of this is to make a copy. So the first thing that we do is we double the DNA and all the proteins, et cetera, et cetera. The next thing that we do is we condense the DNA for easier moving. And while we're at it, we get rid of the walls, right? So we get rid of the nuclear envelope. Then, because we want to put half of everything on each side, we arrange down the middle for easier division. And by the way, one of your checks on the cell cycle happens right here. It tries to make sure that that each spindle fiber is bound, basically, so that you're not pulling too much DNA to one side or the other. Once we arrange it down the middle for easier division, we divide the DNA and divide the DNA. And lastly, we've got to put things back the way they were. So we decondense, so we unpack the boxes, and we add the walls back, the nuclear envelope, and then we pinch the membrane so we have two cells. So I said earlier that telophase and cytokinesis, they often get lumped together, but they're different. Telophase is really related to the DNA, the condensing of the DNA and the housing of it in that nuclear envelope. Cytokinesis is where that membrane is kind of pinching in and dividing. So I want to take a second and give a shout out to our prokaryotic friends who divide by a process that's called fission. And I just want to point out why it's different. Remember that prokaryotic DNA is, one, usually a circle, and two, it's not housed in any kind of nuclear envelope. So that means we can get rid of some of these steps. We don't need to dissolve the nuclear envelope because they don't have one. We don't really need to condense the DNA because it's already fairly condensed. So instead, what we end up doing is we end up being able to skip a bunch of these steps. We don't have to remake a nuclear envelope that we don't need. And really what ends up happening is we double the DNA, we kind of move it to opposite poles, and then we divide the cells. So it's a highly simplified process, and we don't even put these terms, interphase, prophase, metaphase, anaphase, telophase, and cytokinesis on that term. Instead, we just kind of call it this prokaryotic fission, and the only relevant structure is to divide the cells and direct where that membrane pinches in. There's something called a FITZ ring, which is short for filamenting temperature sensitive mutant Z, and that basically just tells it where to separate. If we were in the lab this week, you would be identifying these stages on cells not unlike the ones seen in this picture. This is an onion root tip. And think about it, the root is growing a lot, so it's going to be going through the stages of mitosis. I'm going to be pointing these stages out to you. Most of the cells look like the ones that the red arrow is pointing to. These cells are in interphase. They still have a defined nuclear membrane, and they have the chromatin is not condensed yet. As the chromatin starts to condense, we end up here in prophase. Here, I've moved the arrows to point to the cells that have their chromosomes lined up along their middle, which would be metaphase. In anaphase, you'll notice that the chromosomes are starting to move to opposite sides of the cell. In telophase, the chromatin is moved to opposite sides of the cell, and the nuclear envelope is starting to reform. The chromatin is starting to decondense. Telophase and cytokinesis are often going on concurrently, but you can see here a cell where there's decondensed chromatin and the wall is starting to form in between the two cells. Here's an image of cells from all of these stages together so you can compare and contrast them. Sometimes there are cells that would be in borderline phases, such as late metaphase, early anaphase, and they can be hard to tell apart. So don't get disheartened if certain cells look like they might be in an earlier phase to you.